Anyway, so what we're going to do today primarily is talk about the microbes that live in and on you and me, in and on people. And then the next lecture will be plants with Professor Keene. And in the second Monday section, I assume most of you know, if not all of you, that's when the midterm is. We're going to have a review session today from 4:10 to 5:30, and I'm hoping to have another one on Sunday. Check your email. We should be having one on Sunday also. They will all be in 2205 Herring Hall. And bring questions that you have for me. I'm not going to review like a topic if you just say, I don't understand trees. But you have to have specific questions about something, and I will answer them. And they will be recorded and podcasted, although a lot of the stuff will probably end up being on the board, and we're only going to record audio. So anyway, I will post them as soon as possible. Yeah. Joel has an announcement.
Study for the midterm. Please do a good job, but do not forget about the pre-lab. Due on Monday by 9 a.m.
All right. So what we're going to do today is talk really about symbioses in more detail. And we're going to go through a couple of different aspects of symbioses, in particular a few examples of parasitism, and then talk in more detail about the microbes that live in and on people, which is also known as the human microbiome. So we didn't formally really discuss this in the class, but we've used the term quite a bit. This term symbiosis is used in a lot of different arenas, but the way we formally define it in biology is as follows. So a symbiosis is an intimate association between at least two different organisms in which at least one of them benefits. So many people may be used to using the term symbiosis to refer to interactions where both of the partners in the interaction benefit, but that is actually just one category of symbiosis in the formal definition that we use in biology. So the formal definition basically can be divided into these three different main categories, or classes. So one of the organisms in a partnership has to benefit, so I've put those as organism A here. And then depending on what the effect is on the other organism, we have different terms. So mutualism is the specific terminology used to refer to cases where both partners benefit. So it's thought, for example, that the organelle symbioses between mitochondria in the host and chloroplasts in the host and the secondary symbioses that we talked about, it's thought that those are mutualistic interactions, mutualistic symbioses. And there are many other kinds of mutualistic symbioses. You will hear about a few of these as we go through the rest of the course involving animals and plants and other organisms. There's a second category that you're probably generally familiar with, which is parasitism, which is cases where one of the organism benefits and the other is negatively affected. And we will talk about these, a few examples of these in a few minutes. And then there's a category that is a little bit maybe more unusual or less commonly discussed, which is commensalism, where one of the organism benefits and there's really at least no major effect on the other organism in terms of a benefit or a drawback to the other organism. So these, again, just remember that symbioses is this broad general term to refer to these intimate interactions and not just the mutualistic interactions. So what I'm going to do is talk about one category of interactions that are symbiotic between humans and microbes, and these are forms of parasitism. In the book and in biology courses and in the literature, you will see some slight variants on this terminology for parasitism. And frequently people refer to what are called pathogens. Pathogens are basically a category of parasites. So they are pathogenic interactions. One organism benefits, another organism is negatively affected. So it has the same pattern as the broad parasitism definition. But formally, the pathogen term is usually reserved for cases where one of the organisms is an infectious agent. That is, it can be transmitted by infectious means between organisms, and it causes a specific disease. So there are cases of parasitism where the organisms are not infectious, where you have a parasitic plant that's growing on something else and negatively affecting it. Pathogens are the cases where there's an infectious agent like tuberculosis or flu or HIV, etc. And then pathogens have a lot of different terms that are associated with them. The two most important ones that you will see a lot, both in the book and in discussions and elsewhere in the literature, are these two. So pathogenicity is a very sort of simple terminology, which is basically whether or not an organism is able to enter a host and cause an infectious disease. And then virulence is the degree of the pathogenicity. So the severity of the disease, for example. So if you're comparing two Staphylococcus aureus bacteria, for example, both of them may cause infectious disease, but the symptoms of one of them may be minor, and the symptoms of the other one may be severe. And the one with the more severe symptoms is called more virulent. So it's just a general modifier of the degree of pathogenicity. And again, it's sort of confusing in some of the terminology. You can, in many ways, view pathogen and parasite as roughly analogous, but again, pathogen refers to these infectious diseases as opposed to just any general parasite. So what I'm going to do is take you through 3 examples of parasites, pathogens associated with humans, and talk about their biology and their evolution a little bit. So the first one is the flu virus, or the influenza virus as it is generally known. And the flu virus is a really interesting biological entity. It's very different from many of the from all of the bacteria, archaea, and eukaryotes we've been talking about, and even some of the other viruses we may have discussed a little bit. It's what's called a negative strand RNA virus. For your purposes, what you really need to know is that the nucleic acid, its genetic material, is RNA. And it actually has 8 different pieces of RNA inside this protein shell. Those pieces are called chromosomes, so it has 8 chromosomes. And each of those chromosomes makes, in essence, an individual protein. So this one makes PB2, this one makes PB1. The chromosome names are basically named after the protein that they make. And the most important proteins for what most people care about for the biology of flu are these 2. One is called hemagglutinin, and one is called neuraminidase. And if you see when stories are about the swine flu or some other flu epidemic, the flu viruses are classified by what form of hemagglutinin they have and what form of neuraminidase they have. So the swine flu outbreak was called due to H1N1 flu. That was the different categories of the hemagglutinin and the neuraminidase. And these 2 proteins are particularly important because they make up the majority of the outer coat of this virus. And in general, for most pathogens, the part of the pathogen that's on the surface of the pathogen is the part that interacts with immune systems of things that it infects. They also are the parts of the pathogen that are used to bind to something in the host and get inside of the host generally. So the surface coatings are frequently really interesting to biologists and also highly variable. within a particular type of pathogen, the immune system of a host is going to recognize one of these proteins, and a host will eventually basically develop a immune reaction, either by vaccines or by natural means, and if the virus stayed the way it was, it would eventually be eliminated. But the viruses change, like flu virus changes by mutating the genes that encode the hemagglutinin and the neuraminidase so that these surface proteins change in their structure. So you can imagine the human immune system that's dealing with flu is making these proteins, antibody proteins. You can pretend like they're sort of just like a Y. And you make a lot of different ones, and they scavenge around looking for things inside your body that are foreign, that it wants to destroy. And it recognizes them by binding to particular amino acids and structures inside this little Y part of the antibody. And if the flu virus, floating around, you might have one antibody type in your body that recognizes this. If you get exposed to flu, your body, in theory, if you have a good immune system, will learn and make lots of extra copies of these particular antibodies so that the next time you see that flu virus, you will have many more antibodies and you will be resistant to it. This is also how vaccines work, by developing your ability to produce these particular antibodies that recognize a particular part, usually of the surface, of the pathogen. And each year, the flu virus changes a little bit in the population around us. So if your antibody recognizes the flu virus and you're okay one year, next year's flu virus might be slightly different, a little slightly different form of the neuraminidase and hemagglutinin, and your antibodies may not recognize it quite as well, but they may recognize it okay. And then the next year it'll change a little bit more, and eventually your antibodies that recognize the flu virus from 3 years ago don't recognize the virus from this year. And that's what's generally called antigenic shift in flu evolution. The antigens shift slightly, antigenic drift in the flu virus. They just sort of drift along and change so that eventually the flu virus is no longer recognized by the immune system. What's daunting and interesting and particularly sort of unpleasant about the flu virus evolution is that if you build evolutionary trees of each of these genes, so the neuraminidase and hemagglutinin, and you build trees and you look at them, what you find is that most of the time the genes evolve in parallel, congruent evolution. Each gene is evolving in sort of the same way, and the gene from neuraminidase that infects humans is very closely related that year to the gene from last year's flu strain. And the gene for hemagglutinin in this year's flu strain is very closely related to the gene from last year's flu strain. But unfortunately, every once in a while what happens is an individual, either a pig or a duck or a person, gets infected by two very different flu strains. And the way that flu replicates itself is it makes copies of each of these chromosomes inside the host cell. And if it makes lots of copies of two different forms of those chromosomes, it then makes flu viruses, but it just samples the chromosomes that are sitting around. And you can get what are called recombinant flu strains, where a flu strain mixes and matches the hemagglutinin from, say, a pig flu with the neuraminidase from a human flu. And you get this big change in the surface coating of the flu strain. And so most of the time, the surface coating is changing just a little bit, and the immune system recognizes it sort of. And then 1 year, every once in a while, this happens about every 30 years or so, it's called antigenic shift. The flu coat becomes completely different, and your immune system doesn't recognize it at all. And this is usually when we have massive epidemics of flu, like in 1918 that killed millions of people across the globe, 1968 that also killed quite a few people across the globe. The immune system of the human population recognizes, okay when there are these small changes, but when there are these big recombinations, you get a flu strain that is unrecognizable and spreads throughout the population and kills a lot of people. And of interest, the way that we deal with flu these days is people at the CDC and other places sample the flu population that's out there, sequence genes from the flu population and build evolutionary trees, and try and determine whether or not antigenic drift or antigenic shift is going on or is likely to occur in next year's population. They then predict what the flu strain is going to be for next year, and that's what the vaccine covers, is the predicted flu population for next year based upon phylogenetic analysis of the flu that's circulating around. So, any questions about flu before I move on? So, I want to talk about a second type of pathogen, which is Bacillus anthracis, causative agent of anthrax. It's a member of the Firmicutes phylum, low GC gram-positives. It's one of these members of this group that forms these endospores. Unfortunately, those endospores are very resistant to everything you can throw at them, and that's one of the reasons that Bacillus anthracis is used as a biological weapons agent, because people make spores of anthrax, and then you can store them and spread them around, and they're very hardy, unfortunately, to anything you throw at them and can be used as a weapon. I don't know how many people know this. It's getting to be a long time ago for the age of the people that are in this class. But just after the September 11th attacks in 2001, in October, in fact, of 2001, someone sent letters around to the U.S. Senate building and to some news agencies and to a few other places filled with a white powder that turned out to be anthrax spores. And this ended up killing about 5 or 6 people and ended up causing billions of dollars of economic damage because they had to shut down the Postal Service, the Senate building, as well as many other facilities, and change the mechanism by which letters are screened throughout the United States. The letters were filled with anthrax spores. People opened up the letters, and in fact, sometimes they didn't even open the letters. When the letters were going through mail processing machines, some spores leaked out. And if you are even healthy, but certainly if you have a compromised immune system, a single spore of anthrax that gets into your lung will kill you. So this is, and one letter would contain, trillions of spores. So this was a particularly, scary event for a lot of people. Not going to go into all of the detail behind this, but I was working at an institute at the time that was studying the genome of anthrax before I moved to UC Davis. I worked at an institute that read the genomes of different organisms. Yeah?
How do you kill one of the spores? That's one of the things that it produces to reproduce. It's very hard to do.
How do you kill the spores? Or
yeah.
I'll get to that in a second. What happened was there was a major law enforcement investigation, and the main question people were asking was, can we figure out where the anthrax in the letters came from by doing, in essence, epidemiology of that sample? So just like if there's a food poisoning outbreak, you try and figure out where the disease came from, or with the fungus in the steroid injections that's going around, you try and figure out where that came from. What people wanted to do was figure out where this anthrax came from. And the challenge was really, after some initial assays, it was clear that this anthrax was basically the same flavor of anthrax used in research labs around the country, called the AIM strain. And then what needed to happen was we needed to try and distinguish different AIM strains of anthrax from each other. The genome of anthrax is about 6 million bases of DNA long, and the ribosomal RNA gene, for example, is about 1,000 bases long. These strains of anthrax each had about 6 million bases in their genome, and they differed by only a couple of those letters. So we had to read the entire sequence of the genome of multiple varieties of this AIM strain of anthrax, and then colleagues of mine built an evolutionary tree that was used in the criminal investigation to try and figure out where the anthrax in the letters came from. They weren't able to pinpoint it exactly. You will note a polytomy here, not drawn the exact way we draw them, but it is a polytomy with the anthrax from the Florida letters and anthrax from a couple of other places. So they weren't able to nail it down precisely, but they were able to nail it roughly, closely, which helped in the criminal investigation. So one of the other things that happened and is still going on is decontamination of the places where anthrax was sent. So like I said, and like the question asked, anthrax is very hard to kill when it's in a spore form, and this was part of the major expense. They actually had to seal off the Senate office buildings where these letters were sent, remove the people from them, seal off the Senate office buildings, and then fill them up with a toxic chlorine gas that is, over I think 3 days of filling it up inside these rooms, was able to kill the spores in those rooms, but it's very, very difficult to clean up a particular place if it's got anthrax in it. And this is sort of an aside story exactly how this was tracked down, but an important thing to realize about anthrax is that the reason it makes you sick is it encodes this toxin in its genome. The toxin is actually found on a small extra piece of DNA in anthrax. It's called a plasmid. Many bacteria and archaea have these small little extra pieces of DNA, and it encodes, in some strains of anthrax, they have this toxin gene, but not all of them. And in fact, what people have found out in general across bacteria and archaea is that small pieces of DNA like this, and even occasionally large pieces of DNA, move around quite a bit between strains of organisms by a process called lateral gene transfer. And I'm not going to tell you about this in terms of anthrax, but it is really important in terms of antibiotic resistance across bacteria and other kinds of organisms. So what unfortunately happens in some cases is you treat an infection with an antibiotic. It kills most of the bacteria in the infection, or it might kill all of them some of the time. But occasionally an organism will invent processes that make it resistant to that antibiotic. Say it mutates a particular gene. Those mutations, if you think about inheritance as just being from parent to offspring, aren't that big of a problem. If you just kill the descendants of that organism that inherited that mutation, it shouldn't be a problem. But unfortunately, what bacteria and archaea and many microbial eukaryotes do is as follows. So imagine you have this binary fission process that I outlined before, a single cell giving rise to 2 daughter cells. Occasionally one of them dies, so it gives rise to only 1 daughter cell. And then you have this mutation that, say, makes a strain antibiotic resistant. So all of the descendants of this should be resistant to antibiotics as well. But maybe you can isolate, the individual strain or kill all of it and get rid of it. Unfortunately, what happens is pieces of DNA from this organism can move into other strains by this process of lateral gene transfer and transmit the antibiotic to very distantly related organisms. You can even get transmission from bacteria into archaea, so across the domains of life. So this DNA can move around, and it's nasty when we're selecting for antibiotic resistance in the world, and those genes can move into other organisms. This can create some major nasty problems throughout the planet. It's hard to eliminate individual genes, even if you can try and eliminate individual organisms. So then once this happens, the offspring of this other cell now become antibiotic resistant. And it spreads around the world, not by normal reproductive means, but by this lateral gene transfer process. Now, microbial eukaryotes and multicellular eukaryotes have another means of recombination that you should be familiar with, which is sexual reproduction. So you would have, say these are all different individuals within a species. One of them gets a genetic change. They can produce gametes. Those gametes can fuse and produce an offspring that would acquire the genes from this lineage. So you get mixing and matching of DNA from different lineages in sexual reproduction. The big difference here is the evolutionary distance with which DNA can move. In lateral gene transfer, it can move literally from any organism to any other organism, basically. In sexual reproduction, you basically have this only occurring either within species or among very closely related species by hybridization events. So sexual reproduction and lateral gene transfer create recombinants, but the distance by which DNA can move in terms of the distance across the evolutionary tree is very different. And I'm not going to go through this in detail, but if you're interested, I have a slide with text that compares sexual reproduction with lateral gene transfer and some of the similarities and differences. But what I want to do for the rest of the class now is switch away from the bad guys, the pathogens, to talking about the other organisms, microorganisms that interact with humans. And what you see, if you actually take a microscope out and you collect a sample of, say, scrapings off of your teeth, or maybe your neighbor's tooth if you don't like it to do yourself, or skin, or other places on the body, or fecal samples, is that they are teeming with microorganisms. I mean, literally piles of different kinds of microbes, or different appearance of microbes in the microscope, everywhere you look in and on the human body. In fact, if you count the cells that you see 10 or more times as many cells of microbes in and on the human body as there are human cells on the human body. You know, they estimate for some cases 100 trillion microbial cells, and of many different kinds of microbes that are literally coating all of you. Maybe grossed out by this, but they're there. And so what it's thought for many of these organisms is that they are either mutualistic with us, that is, they have beneficial associations where both partners benefit, or they are commensal in that they're sort of just, they may not affect us really at all. They may benefit by being protected by our gut or something to that effect and stealing food from us in some way, but maybe they don't really have a major effect on us. And so this comes back to the previous lecture. How do we study these microbes? Well, today, and in the last few years, the main method of studying sort of the cloud of microbes that live in and on people has shifted towards more and more DNA-based analyses. And this is using primarily this CSI microbiology approach that I started getting into in the previous lecture that I'm going to go through again here. So the point of this is, if you go to any environment, like a hot spring, or in fact the human gut, you take these samples, you split them in two, like I was saying before, you look in the microscope, you grow things. What you find is many more things you can see in the microscope than you can grow. And for the human microbiome, that's true throughout wherever you look. There are more things that you can see than you can grow. So what do we do in this case? One of the things that we do is to look at the DNA. And again, I started getting into this before. I'm just sort of giving you the background again. So we can go to any environment to do this, but what I'm talking about here is mostly the human microbiome. You take your sample, you extract DNA, you run a reaction that makes copies of your gene of interest, like the ribosomal RNA genes. You read the sequence of those genes, you build a data matrix, and you build an evolutionary tree. And this works if you have one organism that you're interested in. So this is how many pathogens are typed in the human body now. It works if there are two organisms. If there were two there, you would get out two different ribosomal RNA genes, and they'd show up in two different places in the tree. If there were four organisms, you'd get out four different ribosomal RNA genes, and they'd show up in four different places in the tree. If there were 10 organisms, or in fact 10,000 organisms, you would get 10,000 ribosomal RNA sequences, and you would ask your graduate student to build you a tree because you would panic when you saw that amount of data. But in general, this is the approach that people take to characterizing the microbes that are in and on the human body. When people have done this across the diversity of life, they've found some very interesting things. These are those expanded trees that I've been using for a few of the lectures of the bacterial groups and the archaeal groups. I never told you what the colors meant in these trees. What the colors correspond to, each of these are a phylum. Each of these big groups are a major lineage of bacteria or archaea. And in blue are entire phyla for which no one has ever grown a representative from that group. The entire lineage is only known through this CSI microbiology DNA analysis. So this whole window into the diversity of life has been opened up by doing these DNA-based studies. Imagine if, all of the trees on the planet were invisible. That's the equivalent here. In fact, it's probably even higher levels of diversity. Imagine if all of the, Plantae lineage were invisible. and you didn't know anything about them except by indirect means. That's what's going on with many of the microbes on the planet. And you can use this type of approach to do big-scale ecological biogeography, surveying the microbes across the planet, or you can do biogeography of people and treat different parts of people as your landscape. And that's what's been done a lot in the last few years with projects like something called the Human Microbiome Project in the United States and a European project called MetaHit, where people are surveying the natural biodiversity across the human body with these DNA-based methods. And when you do this, you find incredibly fascinating results. So again, I say this every year, but I still can't believe that this colleague of mine felt obliged to use an anatomically correct figure for this paper that he wrote. So I have censored it. If you compare the microbes that are found in different parts of the human body, you find, for example, that across the human body it's dominated by bacteria, at least when you look at just the bacteria, in 4 phyla. So 4 of all of those many phyla that exist in bacteria dominate the human ecosystem. 3 of them you've heard of, firmicutes, actinobacteria, and proteobacteria. There's another one that wasn't included in the 6 that we talked about in the class, but are very important in humans, called the Bacteroidetes. And if you look, for example, at the relative abundance of these different phyla in different sites, in the colon, you find it's dominated by just 2 of the phyla, firmicutes and Bacteroidetes. Yet on the skin, you have an enormous increase in the abundance of actinobacteria, the high GC gram-positives. And you have different relative abundances in the mouth, the esophagus, and the stomach, the vagina, and other places that you look for microbes. And what you should imagine when you're thinking about the human body is each of these sites is basically as different as a tropical rainforest is from a savanna, as the surface of the ocean is from the bottom, the sediment in the bottom of the ocean. Each person is basically a landscape with diverse niches found throughout your body. Some of them are really dry and exposed to the environment. Some of them are really wet and surrounded by lots of food, like the colon, and you get very different microbes in those particular environments. So people have started to zoom in on different parts of the human ecosystem to try and define this landscape in more detail. This is a figure from one paper where they surveyed the skin across all sorts of different parts of the body. You find, for example, that the back of the elbow is pretty different from the, under your armpit. Not surprisingly, at least my back of my elbow is pretty dry. Won't mention what my armpit is like, I guess. But each part of the human body is a niche, and those niches define the environment, and you get different microbes across most of these niches. And again, people have zoomed in and in. They've even compared the right and left side of the hand, different parts of the leg. This poor graduate student in Colorado had every part of his face sampled. It wasn't destructive sampling, it was just a swab, but still. And you get actually different organisms on the top of the forehead from the chin. I mean, this is really basically a planet. Each individual person is the equivalent to the ecosystem on a planet. I've used this term, CSI microbiology, and in fact, one of these studies was used in CSI Miami, not the regular CSI. A colleague of mine, Rob Knight, went and looked at the microbes found on people's fingers, a group of people, and then tried to see whether or not those people would leave their own signature of microbes on a keyboard if they touched it. So you could actually determine, at least at some level of resolution, who was typing on a keyboard by looking at the microbes that they left on that keyboard. So sort of microbial forensic-based studies. So people have done lots of studies like this, surveying the microbes across the human body for a variety of purposes to define sort of what is the landscape in sort of normal, healthy people. And one thing you might ask is, where do those microbes come from? How do we get microbes all over our mouth and our stomach and on our skin and other places? And one really interesting area of research these days relates to the initial colonization of people. So when you are inside your mother's womb, you are hopefully relatively, if not completely, sterile. That is, you have no microbes anywhere on you, unless your mother has some sort of placental infection or some other problem. And during vaginal birth, you get colonized by the microbes in your mother. And in fact, it seems more and more that vaginal birth is partly a delivery mechanism for delivering microbes to the offspring. And you get colonized during that initial phase of your life. You then start to collect microbes from your mother, certainly by touching and breastfeeding if you are breastfeeding, and by other people that you interact with, as well as the environment in which you live in. And at some point, this is pretty chaotic for a little while, and at some point after maybe a year, your communities in different parts of your body start to stabilize somewhat, although it still varies a little bit with diet and with behavior and with health status and with other parts of your biology. So what people are also doing is comparing the microbial communities in and on people that are healthy versus those in and on people that have some disease or some adverse health state. Yeah?
Isn't it true that we get our blood groups from bacteria that kind of surround us in the womb?
Our blood groups are genetically determined, and we inherit those from our parents. But you can have, I don't know if there's any microbial connection, but there can be problems during exposure to the wrong Rh type with blood mixing between the fetus and the mother, but it's not a microbial thing, actually, at all. Yeah?
There's no microorganisms inside the baby. What baby inside the stomach? Like,
Yeah, so when people have done a few studies where they've tried to look at what is in the fetus as it's developing, and there's nothing, basically. So only when you come through your mother do you start to get colonized with microbes. So, for example, people have looked at lots of different... yeah, question?
How do you... What is this is just sort of, I guess, a question where I just want you to guess. How do you think you would feel if you had no microbes on your skin, even for like the briefest of moments? Like somehow you can clear all the microbes off of your skin. How would that change how you feel things versus now?
Yeah, I don't know. It certainly changes your odor, for example. So, you know, the skin microbes play an important part of in our biology. I don't know how they affect sense of touch. I don't know anything about it, but it depends on where they are. There's either thick layers or thin layers. So on your fingers, it's pretty sparse because you use them a lot and things get wiped off. Whereas on other parts of your body that are less, you know, sort of commonly touching things, you might get a big biofilm of stuff on them. I don't want to gross people out too much, at least yet. So, you know, you can compare all sorts of different health and disease states. And one that is an area of great interest these days is looking at just body mass, obese versus non-obese individuals. And like with almost any health state that people have surveyed, there are different microbes found in people that are obese versus people that are not obese. There are different microbes found in people with Crohn's disease versus not Crohn's disease. There are different microbes found in older people versus younger people. There's different microbes across lots of different categories of individuals. It is unclear in most of these cases if the microbes are causing anything, or if the differences are just some effect. When people, you know, are older, they have lots of different behaviors, they've had different history of their immune system, that might just change what microbes are there. People who are obese might just eat differently and change the microbes that are on them, and so on. And what people have been trying to tease apart from all of these studies is, what do differences are there cases where differences in the microbial community actually cause differences in the biology of the host, as opposed to just are the effects of some change in the biology of the host? And so one way to look at this is by looking at what happens when you disturb the microbial community in some way. And so there are many ways that we either purposefully or accidentally disturb the microbial community. One of them is by C-section. So if you are born by cesarean section, you are not getting exposed to the microbes in the mother's birth canal. And in fact, C-section delivered babies have enormously different microbial communities, at least early in life, from those of vaginally delivered babies. And in addition, if you are excessively clean, unlike Pigpen here, when you are a child, you develop a significantly different microbial community from people who are sort of exposed to a lot of dirt. There's something called the hygiene hypothesis. I'll come back possibly to this ailment called necrotizing enterocolitis in a minute, but I just want to talk about another way we disturb the microbial community is by treating with antibiotics. So if you treat with antibiotics, it causes significant changes in the microbial community in individuals. And you can ask, what happens if you have a C-section, if you are excessively clean, germophobic or whatever, or exposed to a lot of antibiotics? Are there any health effects of doing those things? And there are, and they all have what appear to be basically the same health effects. So if you get delivered by C-section, if you are excessively clean as a child, or if you get exposed to antibiotics when you are relatively young, in particular multiple doses of antibiotics, all of these lead to a higher risk of obesity, of autoimmune diseases where the immune system is mistakenly killing parts of your own self, as well as a variety of inflammatory problems like allergies and other inflammation-related diseases. One theory for these symptoms is that each of these disturb the microbial community early in development, and that leads to some miscommunication with the immune system. So the immune system during development is not only trying to recognize the parts of your body to not kill the parts of your body, it also has to recognize all the non-pathogenic microbes in and on you and not activate an immune response to them. And if your microbial community is thrown off, it appears that your immune system does not develop quite correctly, and you end up with an overstimulated immune system that leads to things like autoimmune diseases, type 1 diabetes, for example, or all the other autoimmune diseases, as well as obesity, which appears to be, in some cases, an inflammation-related symptom, and allergies. Now, it hasn't been proven in these cases if changes in the microbial community caused these things, but it is very suggestive that if you create all of these different disturbances, the thing they have in common is affecting the microbes, and what they do is cause problems in the development of the immune system. So this is one of the classic pictures and studies of microbial communities. This is, it may not look like it, but this is a mouse. This is a mouse with a genetic mutation in a gene just called the OB gene for obesity. If it has this mutation, these mice become obese. This is a normal mouse. So here's an interesting thing. You compare the microbial community in these two mice. The microbial communities are different, just like in obese people versus skinny people. But here's the amazing thing. This obesity is probably caused initially by that OB mutation. But if you now take the microbial community out of this obese mouse and you inject it into this skinny mouse, it becomes obese, even without the genetic mutation for the obesity gene. So the mutation leads to a different microbial community, and that microbial community can now transmit obesity to another organism. So this study alone has launched a lot of people into trying to figure out what are the effects of the microbial community on phenotypes like obesity, inflammation, autoimmune disease, etc. And so the last thing I want to talk about is, so you disturb the microbial community, either on purpose or by accident, or you diagnose that someone has a disturbed community. Even if you don't know what caused it, you can compare people to healthy people and possibly flag individuals as having an unusual microbial community. And let's say you want to restore their community to something that you consider sort of more healthy or potentially more healthy. Now, I assume many people are familiar with probiotics. This is what many people do to try and modify or balance their microbial community in some way. Probiotics come in pills. Obviously, there's lots of yogurt and other fermented beverages and now fermented foods and other things that are thought by many people to be good for you because they bring in beneficial microbes into your system. There's not a lot of clinical data to support most of those claims, by the way, although there are some. So there are well-established cases where adding individual probiotic microbes into your system is beneficial to your health. So this has been shown, but not for most of them. But what you're doing here is basically the equivalent to, let's say you're trying to restore a tropical rainforest that someone cut down. This is like planting one species of tree to restore the tropical rainforest. It's not the most elegant, best thought out way to try and restore the microbial community. And here's, in the last few minutes, the gross part. So what are people doing instead? They are doing what are called fecal transplants to try and restore the microbial community. Yes, they are doing this. To try and restore the microbial community in people. This has been done on and off for about 100 years in various places. It is routinely done in animals, which we can talk about some other time. Yeah?
I was going to ask, isn't it not, I remember reading an article about this last year, isn't it not approved by the CDC or something? Or not some organization, the AMA or something?
Yeah, well, we'll get to that in a minute. So this is nevertheless being done. And what is being done in most cases in the United States right now is treatment for a particular problem that, for example, this woman had. So many people get what are called these persistent Clostridium difficile infections, or C. diff. It's a firmicute bacterium that forms spores and is very hard to get rid of infections with this in some individuals. They can have diarrhea for 20 years. They can die from this. I mean, the persistent infections will kill people. And there appears to be no way to get rid of them with antibiotics, for example. But if you take fecal matter from a donor and you clean it up a little bit and you deliver it to a recipient, such as through a nasal tube, hey, the last class had to have this before lunch. And you deliver this through a nasal tube, there is estimated to be about a 95% success rate in curing C. diff infections. So what people are now exploring is doing this type of fecal transplant for many other cases where the microbial community might have gone wrong in some way. And you will see much more of this, for better or worse, in the next few years. Just as an aside, she got the donation from her mother. Just wait 1 second. What's going to happen is people will grow as many microbes as they can in culture in the lab and deliver them by much more pleasant means than by a fecal transplant in the long run.
Community. So wouldn't it make sense to get donors that are closely related? Because
it might, but it's not clear yet.
It's not clear, but how much? It's kind of like a better safe than sorry.
Yeah, probably.
Okay. All right. Well,
Question about the lecture. So is there a difference between the